So today we're going to talk about next generation sequencing, also known as NGS. And so in this lecture, we'll walk through how sequencing technology has evolved, how Illumina sequencer works, how the data is processed, and then also different type of data formats so that you can learn how to interpret different next-gen sequencing output. So we're talking about formats such as FASTQ versus a BAM file versus a SAM file. By the time we finish this lecture, hopefully you'll understand how wet lab sequencing connects with how you do the computational analysis. So in this lecture, we're specifically going to look at 4 different topics. We'll cover DNA sequencing and talk about its evolution over time. We're going to talk about next-gen sequencing technology. Specifically, we're going to look at Illumina's NGS approach. Illumina techniques, we're going to dive a little bit further into experimental design and some more information on NGS technology. And the last thing is, how do we go about doing data processing of these NGS sequencing data? So how do you go from raw reads to a format where you can either study expression, study transcription, or be able to do variant calls? So let's first talk about the evolution of DNA sequencing. So the first sequencing effort was actually started with RNA and not DNA. This is in the early 1970s. And in 1977, the Sanger group developed a way to do chain termination method that allowed them to be able to sequence DNA. And this became the gold standard for many years. Sanger sequencing was able to provide long, accurate reads. So it was really good for us to be able to look at different regions that are maybe de novo sequencing or highly rearranged regions, such as in cancer genome. And then we progressed and started moving into massively parallel sequencing. So this is when we started getting into next-gen sequencing. This allows for millions of DNA fragments to be sequenced at the same time, hence the parallel sequencing. We are now entering the 3rd generation right now. And so the 3rd generation is also known as long read sequencing. So as I mentioned, sequencing was first taken on RNA, right? So RNA was the first sequencing that we got. In the 70s, we're moving into DNA sequencing. The Sanger groups developed something known as Sanger sequencing, and it was used for many years until we moved to the 2nd generation, which is when we were able to start doing massively parallel sequencing. And so that was in the early 2000s. And now we are able to do large-scale sequencing, but we're able to do long read sequencing, which is the 3rd generation. So how does Sanger sequencing work? So Sanger sequencing is also known as the classical chain termination method. So using this method, what you have in any reaction is you have a single strand DNA template, you have the DNA primer, you have the DNA polymerase, which is the enzyme that's going to allow us to elongate from our primer on, and you have normal deoxynucleotide, but you also have this modified D-deoxynucleotide that is lacking a 3' hydroxyl group. So when it's incorporated, what happens is it ends up terminating the DNA strand elongation. So in this method, you're going to run 4 reactions, 1 for each ddNTP and the remaining dNTP. The concentration between the ddNTP and the dNTP is typically 0.5 millimolar of the normal dNTP to 0.005 millimolar of the modified D-deoxy NTP. And so you want enough fragments to be produced while you're still transcribing the complete sequence. But we also want to have a few of the fragmented and early terminated strand as well. So following each round of template DNA extension from this bound primer, you're then going to have the DNA fragments at the end be denatured by using heat, and then we're going to run it on what's known as a gel electrophoresis that allows us to separate these different templates by the length. Okay, so to recap, right, we're going to have 4 reactions. One for each nucleotide, where you have that modified version of the nucleotide and 3 of the normal remaining nucleotide. So you're going to synthesize the DNA fragments in these 4 reactions. The fragments are then heated and denatured and separate into this gel electrophoresis. So now they're going to run this gel electrophoresis that's going to be able to separate them based on size. And so you have one for each of your reactions. Remember, because you have one reaction for DDATP, one for DDTP, and so on. And because these DNA fragments are labeled with some sort of tag on the primer, right? Some kind of either radioactive or fluorescent tag in the primer, we can then be able to determine the sequence of our fragments based on the result of the chain termination after the DDNTP has incorporated and the DNA synthesis has been terminated. So with time, technology allows us to automate single DNA sequencing. So what we see here is that we have a reaction mix. It has the same component that we mentioned before, the template, the primer, the enzyme, and then the DDNTP that has a fluorochrome, and then additionally the normal DNTP. And so what we can see is that with the primer and the template and the different DDNTP, we can then run in this reaction, and instead of having to use a gel like before, we can actually detect it. So with the gel, typically you had to do a visualization of the gel after you ran it, right? Using either some sort of dye or using, in this case, during that time, they mainly use UV to be able to actually detect the bands on the gel. But now with the fluorescent, we can then take the fluorescent as a way to be able to determine what that last nucleotide is, and from there guess what the sequencing is of your template. So here we take all of those fragments, and it's going to run it through this capillary gel, separate them, and then be able to read the fluorophore that's at the end of each one of these fragments to be able to detect what the sequence is. And similar to what we saw when we were looking at the matrices and the motifs, is that whatever the highest peak is, that's the one that we're the most confident in with regards to if it's a D, I mean a T, a C, or a G. Now, one of the things I want to mention is that even though this method is pretty accurate, it's very low throughput. That means you can only get one sequence per reaction. So later, when we look at next-gen sequencing, often you hear it referred to as high throughput sequencing, and it's because we're able to run parallel sequences. So you're getting more than just one sequence per reaction. This method, though, once we were able to automate the singular DNA sequencing, sped up the reactions much more dramatically, but we still couldn't scale it to be able to read a whole genome efficiently. Okay, so let's move on to next-gen sequencing technology. So now we're moving into the second generation sequencing. So in this second generation sequencing, we're able to sequence at a much higher scale at this time. So what we have our DNA sequence that we're interested in, and we're going to fragment it. And this fragment happens relatively randomly. So each sequence is going to be about 300 base pair. The reason why I say relatively is that depending on the type of method that you use to fragment it, some method has a certain bias toward fragmenting it at a certain point within the sequence. And so that's something to just kind of be mindful of, is that even though a lot of our methods we use random in biology, oftentimes there's a certain bias that maybe we didn't account for. And so I'll bring that up again when we talk about amplification, right? So when we amplify our DNA, RNA polymerase and DNA polymerase at times can have a certain bias to them with regards to which sequence they prefer to elongate or to replicate compared to other ones. And so downstream, when you're doing your analysis, that's just one of the things for you to take into consideration, is if you have a lot of certain sequences that you see many duplicates of relative to all the other sequences. Okay, so we have our DNA, we fragment it, and now once we have the fragment, how do we go about amplifying these fragments? Well, there's two methods that you can use to amplify. We can either do in vitro cloning, which is the case where we take our fragments, we place it into these plasmids, and then we create multiple copies of the DNA fragment before we sequence it. So we create multiple copies of these, and we grow these up in like bacteria, for example. And so you have each of these bacteria will carry a plasmid or multiple plasmids, and you will grow that up, and then you will isolate this plasmid out. And that's one way for you to be able to amplify. Another method is to massively parallel, do massive parallel sequencing, also known as MPS. And so this allows you to take the sequences and you're going to make multiple copies of that strand simultaneously. So let's move on to how we would do this commercially. So in commercialized NGS, and we're going to specifically be focusing on the Illumina's approach. Approach is really similar, where what we do is we take the genomic DNA, also known as gDNA, right? You take the genomic DNA, we're going to fragment that DNA into a library of many small little segments right here. We're going to amplify those segments and sequence them in parallel. So now we'll take millions of sequences and we do parallel reaction. And then once you have them parallel, you end up with what are called reads. So the reads are the sequence segments, and you're going to align them to some sort of scaffolding, so some sort of reference genome. So we do the alignment, and then from that we can pull out what our consensus sequence is. There are times in which you do not have a reference genome. So an example with this would be a certain species that is studied in biology, but not that many people are interested in. So an example of this is the lab that I knew in over at CU Boulder. They were trying to understand how metabolism occurs in python, the snake. But the python snake didn't have a fully reference genome. So a lot of the alignments, as they got these sequences out, they had to do de novo sequencing and try to align all the sequences and guess what the reference sequencing is. And so the more experiments you do, the more sequences that you can start gathering that will overlap one another, and eventually you can build a full reference genome. But sometimes you're lacking that, in which case you're doing de novo sequencing. Okay, so let's go into the Illumina approach a little bit more detail so you can kind of see how we go about doing the wet lab component of next-gen sequencing. So before we get to this step, we had done our experiment, right? So you'd done your experiment, you collected your samples, and you took the cells and you open up the cells, you extract the RNA or DNA, we're going to assume is DNA. We extract our genomic DNA, and in order to start preparing your library, you're going to have to take that genomic DNA and you fragment it. So that's this step one. So sonication is one of the methods that you can use to fragment it. There are more than this method in order to fragment it. We're just going to focus on sonication. And what sonication does is it basically applies a sound wave that will actually break up the genomic DNA into smaller fragments. After you have the fragments, you need to prepare the end, because the fragmented will produce these fray ends right here. So you're going to do some end repair. And once you have the end prepare, then you want to bind onto your DNA some adapter. So you're going to ligate. Ligate means kind of like gluing them together, right? You're going to glue these adapters to the DNA end. And sometimes we glue them to one end of our DNA fragments, and sometimes we glue them to both ends. If you're gluing it only onto one end, where you're preparing what's called a single end library. If you're gluing it to both ends, you're going to produce a pair end sequencing library. Once you've done the adapter ligation, then you just want to check your samples and do a QC check. And this is basically to see if you have enough concentration of your fragments in order to actually be able to get some data out of it. And you also want to check if your fragment size is within a good length. So you want the fragment size to be roughly 100 to 500 base pair length. You're going to get a few that are really small, you're going to get a few that are longer. If they're too small, you might not be able to get enough information to be able to get a read that you can align and produce any real detail, because remember that we have the adapter sequences, and then after that, the first few sequences that are going to be sequenced later on in the next few steps are always a little bit lower quality, and we'll mention that again later. So if you have a fragment that's very short, it's probably going to be poor quality. If you have a fragment that's really long, it's going to be really hard to do the post-processing steps when you're getting to the point where you actually have to align the fragments to the genome and so on. So you're going to run this QC check next in your library preparation. After you have your library prep, then what you're going to want to do is you want to now amplify your library prep. So we're going to take all those fragments that we have with the adapter aligned to it, and we're going to put them on what's called flow cell. So here's your flow cell right here. And so the flow cell is going to bind these single strand DNA onto flow cell. And the flow cells also have adapter onto it. So you can think of an adapter as basically they're going to be complementary to the adapters that you had ligate to your DNA fragments. So because they're complementary, this DNA single cell is going to find an adapter here, and it's going to be able to form a bind together. And once you have the fragments on your flow cell, what you're going to do next is it's going to produce what's called a bridge. And so it will start elongating, folding over in order to find the other adapter. So you have an adapter here, and it's going to form over, it's going to bind to this adapter over here. And so you form what's called a bridge. And once you have this bridge formation, that's when you start doing amplification. So we're going to start having the strand start elongating right here. So we're going to form an amplification. And once it reaches onto the side, it's going to get back into this type of formation again, and it's going to form the bridge again, and it's going to keep repeating that until in the end you have this set of clusters of single strand DNA. Why do we want a cluster versus one? Well, we're going to use these DNA later to do the sequencing. And so we're going to use fluorescent in order to be able to detect that sequence. So if you have this whole flow cell and you only have one of these strands lighting up, it's going to be really hard for the computer to detect what that fluorescent is. But if you have multiple single strand DNA here that are all going to amplify the same color, then the machine can be less sensitive and still be able to detect whether or not the sequence that it's reading is an A or a T, for example. Okay, so you have these clusters. Now, how do you actually go about doing the sequencing? Well, once you have the cluster, you're going to start adding the DNA polymerase, so the same one as we saw in the Sanger sequencing, and we're going to have a bunch of label dNTP. So these NTPs are labeled with fluorescent. And what's going to happen is that it's going to start replicating here, and we're going to do what's called a base calling. So what's happening here is you have the dNTP incorporating onto the elongation. A laser is going to excite like all the different strands here, and that's going to excite the fluorescent. And so when we do an image capture, it can capture what that fluorescent is. The tag is then removed, and we're going to repeat this method again where you add another dNTP, you excite it with the laser, you capture the image so that you know which sequence it is or which nucleotide it is, and then you repeat. This is called sequencing by synthesis. And you repeat this sequencing cycle, and then you can also do what's called dual flow cell, but we won't cover that topic too much. So one thing to keep in mind when you're doing your prepping of your library and you're doing the quality control step is your concentration. If you have, when you're measuring how much nucleic acid you're starting with, how many single strand DNA that you're starting with, if you have too little, you're probably not going to be able to get a good library because it's hard to get enough coverage if you only have a few fragments, right? It's hard to be able to get enough of a coverage of that area because remember, the fragments are smaller area. Let's say your gene is like 20,000 base pair. If you only have so much sample of DNA sequencing, you might not be able to cover that full 20,000 base pair region. If you have too many DNA fragments and your concentration is too high, when you hit this step right here, or actually I should say when you hit this step right here and you're loading onto your flow cell, you might end up with clusters in the end that are really, really close to one another. And so if you have these clusters, if you have a flow cell that's too dense and has too many of these clusters, then what's going to happen is when you get to this step where we excite and we're trying to do base calling, we excite the cluster with our laser, is that if they're too close to one another, how can you really distinguish if that was a red signal or a green signal? Like what if they kind of combine on top of one another? Like let's say you have a yellow and a green signal, then you end up with like this bluish color, and it's hard to distinguish was that a blue signal or a yellow signal. So another thing to kind of keep in mind is your concentration before you even get to the step of sequencing. But all the way back to here when you're doing your quality control check. Because remember, similar to what we've mentioned before, if you put in a lot of garbage, in this case it could be the fragments are really short, or you don't have enough depth, or it could be multiple different things that makes it to your library is low quality. You can still run through all these reactions, but then when you actually get to this part and you're getting your results back, and now you're trying to actually make something meaningful of it, it'll be a lot harder if you have low quality to begin with. Okay, let's move on to the next step, which is distinguishing again what the difference between single end and pair end sequencing is. So you have your fragment, and if you put adapters on both ends of your fragment, you will produce what's called pair-end sequencing. So pair-end sequencing means you have fragments on this side and on this side, right? You have adapters that you add on both sides. And what's different between it versus single cell is single cell you end up with just this read right here. So I might not have made it clear when we went to this step right here, is that you have adapters on both ends. That's not exactly the reason why you would call it a pair end. It's pair end right here because we have one read where we have these adapters, we'll just call them blue adapters, on this 5' end side, and when we have the same blue reads, blue adapters on the 3' end. And so because you need to actually have adapters on both sides because you have to have adapters in order to form this bridge formation. But because in pair end you have two sets of reads covering that same fragments, you have this we'll call read 1 and this which we call read 2. What's going to happen is that you'll have elongation that goes both this way as well as this way. And so later when we try to do our alignment, you'll see that if you have both ends, you're more likely to cover what's in the middle. So keep that in the back of your mind, and let's follow up on that when we have a picture. It'll be a little bit easier to visualize when in a later slide, and I'll point it out then. Okay, so this is an example of what a flow cell might look like. So the flow cell will have these lanes. And so these are the lanes right here, and I'll have a pair of lanes. So this is lane A, and this is lane B. What you're going to do is you're going to place your library, your sequencing library that you prepare, and you're going to throw it onto this flow cell. This flow cell is this glass-based substrate that is going to be where the sequence clusters are generated, right? So when we looked at this image right here, this is our flow cell right here. So we have this glass base with a bunch of adapters on it that's going to allow your library to bind to the adapters, and then you can do the sequencing reaction on these in these lanes right here. So the lanes are these small imaging tiles, which is where we're going to excite the fluorescent with the laser and be able to do the reading. The library is what you're going to place onto this flow cell. So once you've done your sequencing, what you end up with is this set of reads, right? So here's your flow cell. We've done this part. We formed a bridge. We got the cluster. We took the clusters. We did our base call. And so now, after you do your base call, you end up with these reads, which are all these little fragments of sequences. So once we have these fragment sequences, we need to map it. So what we mean by mapping it is that we're going to take these fragments and we're going to refer to a reference genome, and we're going to try to align it to this reference genome in order to form a map. So by taking all these sequences, and again, your depth is the amount of reads you have. So if you don't have a high concentration, you'll end up with just like 1 or 2 reads covering each one of these positions, right? But we need more than that. We want enough reads so we get like quite a few of a pile up in areas where we really need to know what the sequence is, because we want to make sure that we have consensus across all of these reads at each particular region. So you're going to have all these reads, and you're going to map it to your reference genome, and you'll end up producing this consensus read. So see how here we kind of have these reads, and we're trying to build what the consensus would be between them. But you have these reads here that came from your sequencing library. So here, what you see is a bunch of these reads. And so you have a bunch of these reads. We're piling them up, and you can see the pile up here. This is IGV. So this is known as an integrative genomic viewer, and it's a way for us to be able to visualize our alignment. And so what you have here is you have your chromosome here. This is your reference chromosome, and you have your base pair here as far as the position. And at each of these positions, how tall this peak is for example, here, how tall this peak is how much coverage do you have? How many sequencing read do you have that's covering each position? So this right here is also known as your depth. Down here is your reference genome itself. And so at each position, we're going to look and ask what is the sequence in each one of these reads. If it matches the reference, it's going to be gray. If it's not matching reference, it's going to be some sort of color. And so you can look at each position. You can zoom in and out of this to look at each position to determine what the sequence of the nucleotide is at that position. So let's kind of summarize what we've gone over. So next-gen sequencing technologies we mentioned is a method that we can use to do sequencing run. And we can get a lot of details about the sequencing run depending on our concentration or our quality of our reads so that we have enough coverage to get what we call a good depth. So coverage is an average number of sequencing read that you have that aligns to each base within a sample. If you have a good deepish sequence, right, on average the sequence is 50x, but let's say you have an area that you really want to sequence in deep, then you might do like a higher amount, like 100x or something. And that would mean that on average, you expect that each one of these bases in the genome is covered by around 100 sequencing read if you have 100x. The average is typically about 30x. There are certain times in which you want to sequence a lot higher depth. Like let's say if there's a mutation that only happens in a very small subset of cells, or if there's a very rare mutation, or if it is a certain region that you only really see amplified or expressed in a certain disease progression, in all these cases you would want a much higher coverage, because if it's a rare position, it's even less likely to be touched, and so you want to make sure you have enough coverage so that you can actually get the actual sequence from there. Okay, so the next thing we're going to talk about is next-gen sequencing data processing. So coming back to this is basically what we're going to talk about now, is that now you have all these reads, and these are all these different fragments of this reference genome that you have interested. And you need to figure out how to figure out where all of these different reads align into this story. So you're going to use a reference genome so that you actually have an idea of where they might align, and then try to do the alignment. So this is pretty much our next-gen sequencing data processing workflow. There's a lot more to this, but as a whole, this is what we're going to kind of observe, where we take our raw reads from our sequencer. So this is the sequencer here. It does the base calling. You get back what's called FASTQ file, and you're going to want to align the FASTQ file, and you'll end up with what's called a SAM file or a BAM file. You then take that SAM or BAM file, you do a few more things to it, and then from there you can decide what it is that you want to do. So do you want to look at transcription? Do you want to look at variant call, for example? So let's say you want to look at variant call. You want to see the difference between person A, person B, and person C, and you want to be able to look at certain region to decide how often you see gene X, different versions of that gene X. So you could do something called a variant calling. So let's go into these in a little bit more detail. So the first thing off your sequencer, fresh off the press, are these FASTQ file. And a FASTQ file looks like this. So what you're going to get is you're going to see multiple lines. Each one of these lines represents a read. So you have these raw unaligned reads. So right now you haven't done any alignment. These are just your raw reads. You're going to take these raw reads and they come in blocks. So each time you see an @ sign here, that is the beginning of a block. Each block has 4 lines to it. The lines are the identifier or the gene name. So this right here, depending on your sequencing, usually is the name of the read, and it will tell you like what lane it is, which flow cell, which flow cell did you use, what lane was it in, etc. There's like other information that can be detailed in there. Your next second line is your sequence themselves. Your third one is optional, is typically a plus sign. And then the last one is the quality of your bases. So notice how they're the same length. So it goes through and it tells you the quality of how likely it is that it is correct as far as calling that base. And each, again, your FASTQ will have multiple of these blocks. So each time that you see a @ sign, that's the beginning of a block. So this is one read here. This is a second read right there. Now you actually want a read that has good base. And so we're going to use what's called an ASCII table right here to look at the quality of our base, also known as the FRET or the Q score. Basically, this is asking how confident are you in your base call. And so this table I find to be a little bit easier, and you can actually link it right here and see it. So you'll have a quality score here and the FRET error right here as far as how confident you are in a quality that you correctly call that quality, that base, sorry. So the FRET scaling is basically, this is how you go about calculating it. It's minus 10x to log of 10 of the error rate. So if you have a quality of 10, for example, then basically you look at the error rate of 0.1. So if you go back to here, you go to quality here, and you can see that your P error rate is 0.1. What that means is that you have a 10% chance of error right there, and you're 90% confident. The higher your number, the higher your confidence, and the lower your error rate is. So a Q20 with a FRET score 20, you're going to be even more confident about that base calling and have less of an error compared to 10, and then 30 will be even more confident. Often we're using a FRET 33 scaling. This just means the SAM, the file, starts encoding by adding 33 to the value, and that's the first character, and it moves from there. So here's some more information about your FRET calling, right? So here's your FRET score. This is the probability of incorrect base calling, and this is the probability of the accuracy. So the higher the FRET score, the more accurate it is. And so here you have this quality, you have your base call here, and you can kind of even visually see as you are right here where you have a little bit higher FRET score probably, you can see that it's pretty confident in what it's calling. As you get to here, you have more N. N is typically means that it doesn't know. It can be any of the NTP. It can be an A, T, C, or G, and it's trying to guess. Here, even though it's calling it A, you can see that in many of the reads, it calls it as an N. You also have some that will be calling G, as a red. I don't remember what the red color stands for. The red would be the T. So you see that sometimes it calls a T. So this FRET score is going to be a lot lower here compared to an area over here. Okay, so you have your FASTQ file. One of the ways you can determine, right, because it's really hard to actually go through this and try to interpret. And remember, in every reaction, you're going to have millions of these reads. So you're going to have millions of these reads. For you to go through and try to figure out read by read, that's just ridiculous. That would take forever. You won't be able to do it. So we have these different software that allows us to do a quality control. So FastQC is one of them, where it's a tool that allows you to do a bunch of quality control. So what's happening is that you have your FASTQ file, and you can run it through all these different programmings, right? So first thing you're going to do, for example, is aligning it to your reference genome. You're going to do trimming, which we'll mention a little bit later, but it's going to take all of the different tools that you're going to use in your pipeline and it's going to take feedback from that in order to produce these quality control report for you. So this link right here, let's open it up real quick. So this is the website that allows us to actually see the FASTQ report. So the FASTQ, like I mentioned, takes a bunch of output file from your FASTQ file itself, and it does this quality control. And so we're not going to really run we're going to run a FASTQ, but I just want to point out a few things so that you know whether or not your data is good data or bad data. So this is an example of a FASTQ report. This is what the FASTQ report will look like. And here you have your good data. So it's going to go through each sequence and it's going to basically give you back a quality score. And basically you want the quality score to be up here, right? We want a higher quality score because it means that we're more confident and there's less error rate. If you start having your quality of your reads come to the red level, even the yellow level, that's a sign that that's a bad thing. This report will also tell you some information such as what is the sequence length, each of your reads, and then your reads, what percentage they are. So we scroll down. This is your actual flow cell. And so basically this is good right here, all blue. If you start seeing dots of colors of tiles and they're different in color, that's usually a bad sign. That means that in your flow cell, you have some bad data. This is your per sequence quality score. So this is the quality score on average. So on average, you have a pretty good quality score, right? It's about a Phred score of about 37. That's a good thing. This is the GC content. So this is going to be dependent on each species. They're going to have a specific GC distribution. But you basically want to see one peak here to show that there's really only one species. And this is your length. We want all the length to be roughly the same size. This is your sequence duplication. We don't want a lot. We want each of the reads to be unique, right? So we want the peak to be here in 1 because we want multiple sequences that you only see one copy of. If you had a peak like even here, but if you had a peak that was like really high right here, that's pretty bad. Like you should not have multiple reads that show up more than 1,000 times. So that's kind of what you're looking for. So this is your adapter content. So this is what a good FASTQ would look like. Okay, let's look at what a bad Illumina FASTQ would look like. So this is what a bad one would look like. Okay, see, these ones look pretty good, right? But notice how, oh boy, we got into the red and yellow here. This is a bad sign because now you have all these sequences that have a Phred score of like 2, 4, 6. This is not good. This is not what you want to see. And see here, you have all these colors. This is your flow cell, right? So your flow cell, if you see a bunch of these colors here, that's a bad sign. That means that it produced poor quality here. And we want it to be full blue as the way we showed before. See your quality score. See how you have average qualities here, but you also have like this big bump right here. This one looks okay. What was this one? Per base. You want this to be pretty much a straight line. As far as the end content, you don't want a lot of end content. This is roughly the same sequence distribution. See, we don't want a lot of sequences where you have like over 1,000 version of it. And this is telling you all of the overrepresented sequences here. These sequences could be, they could be coming from your adapter. They could be coming from, like you should have certain sequences that are like really duplicated. Like during your PCR amplification, you had a bunch of sequences that were like duplicated for some reason. It can come from multiple different sources. Adapter content looks funny. Okay, so this gives you an idea of what a bad one would look like. So you typically want to run a FASTQ-C report so that you can see the quality of your reads. Once you see the quality of reads, one of the steps that we typically want to take is filtering and trimming our reads. So we want to only bother aligning reads that are good quality, right? We don't want to align just any reads. So we want to filter your sequences to remove the ones with really poor quality score. Reads are too short. Reads that have way too many ends in them, like it just didn't do a good job of base calling. And then maybe we had a contamination, so we want to move reads that have like a really high, for example, GC content. Additionally, you want to do what's called trimming. So here's our fragment right here. There's our fragment, it has a 2 adapter on each end. So you can kind of imagine that if you have your fragments and you have these adapters, and these adapters are specific to aligning and binding to your flow cell, right? But they're not found in the genome. So we want to get rid of these adapters and trim for them. Additionally, oftentimes you want to trim the very end right here and right here, because as you're reading through primer, when you're first going through it, oftentimes these end pieces are a little bit lower quality compared to in the center. So sometimes we want to trim the beginning and the end of the sequences because the quality is not, there's more sequencing error basically. So we can trim our data. So for example, here you didn't trim your data yet. And so you can see the quality looks pretty bad. But then once you actually do the trimming, your qualities improve. So the software tool that we're going to look at is going to be Trim Galore. There's a lot of them. There's Trim-O-Matic, there's bbDuck, there's quite a few different programs. Similar to Alignment, there's pros and cons to each program depending on what it is that you want to do, right? So we're just going to do Trim Galore because that's what's available in Galaxy, and that's what we're going to use to do some of the dry lab part, but there's many more. Okay, so now that you've done your alignment, you do the trimming, I would recommend that after you do your trimming to run FastQC again just to see whether or not it improved the quality. But once you have that, what you're going to get back is this SAM file. So this SAM file is, what's nice about it is that it is human readable. So it's this uncompressed text file that is human readable. And so we can actually take a look at it and understand it. I think the next slide I have a picture, right? So here's an example of a SAM file. So you can actually get the quality name. You can see the flag of it. It tells you the position, right? This is the second line right here. Like all of this information is easier to understand. The bad part is that because it is human readable and it's uncompressed, it's really big. It takes up a lot of space. Remember, we're dealing with millions of reads here. It takes up a lot of space, and a lot of the downstream program doesn't work well with SAM. They want to work with a binary output file. So you would take your SAM file and you would convert, whoops, this is supposed to be file. And you want to convert to your BAM file. Your BAM file is compressed and is indexed. So it uses an index to be able to sort it. And it's also binary. So it's not human readable to us. We can't understand what it's trying to say. So let's open one up just so you guys can kind of see it. So here, if I go ahead and just cut my BAM file. Yeah, so I can't understand what that's trying to tell me. Oops. Oh. But I can't understand what that file is trying to tell me. So that's what a BAM file is. But a lot of the downstream program that you're going to use can actually understand that file. So kind of coming back to this slide, after that you can do different steps. So you can do VCF to do variant calling, but you can also do multiple different things such as DESeq to do expression calling. So there's multiple different things that you can do with it. We're going to talk more now about, the next step is about where do you get your actual data. So if you are interested in actually finding sequencing data you can work with, and you're not going to do the wet lab yourself, like I mentioned before, there's so much data out there, One of the things you can do is you can go to the Sequence Read Archive. This is a database that is just full of biological sequences that's available to the research community. Most of this data was produced by different labs that have experiments. They're publishing, and then now they're placing it onto this public repository so that you can have access to it, so you can sequence it. So you can find everything from raw sequence file, such as the actual FASTQ file, but you can also find some files that have already been aligned and has some processing. So for example, previously we had worked with that chip file, and if you kind of recall, I said that this chip file was an output of the max calling. So they've already aligned it. They've already called the peaks for where they think they see the enrichment of the chip sequencing, and so you can get a data that already has some processing done to it, or you can actually pull raw sequences. Once you have your raw sequences, right? So you put in here in the field a certain you can use a session number of a peer research that you're actually interested in. You can be more broad and just say you want to find work that's RUNX1 in mouse or something, and it could probably find some files for you. You're gonna land on a page that looks like this, and you will see that it'll have a SRS number associated with that biosample. And if you click on into it, you can, so this is the SRP, this is the a study. So this will have all the sample. If you click onto this, it will actually open up so you can see like multiple different files that are associated with that study. And so you can click on each one of those. Once you have those files, you can either use an SRA toolkit to be able to pull down that SR file. You can do it on the terminal as well. So we're not going to do this because many of these files are really big. So there's only so much that we can do on our local machine. But this just allows you to know. So if you do have access to a server and you use a server, you could then use, like, for example, fetch. You can use this line right here on your terminal where you do a FASTQ dump, and it will download the compressed zip file for you for this study, this SRR, for example. And so then from there, you can have it on your machine or in your directory in your terminal, and you can start processing it more. So I'm going to end the lecture right here. And what we're going to do now is we're going to actually play around with understanding how to actually get data, and then from the data do some quick quality and some quick processing with it. So we'll do a FASTQ, we'll trim the reads, and we'll align it to our reference genome. Okay, I'll see you guys soon. Bye.